Hi, my name is Jessica Hansen. I am going to be discussing the Unit 4 case evaluations, cases 1 and 2. First, for case 1, we start by discussing the normal mechanisms of free amino acids. Usually in the muscles and the liver, they will undergo a reaction called transamination. Transamination is when the amino acid, let's say alanine in this case, is interacted with alpha-ketoglutarate. This interaction uses alanase aminotransferase, or ALT, as well as a PLP cofactor to make a new keto acid, which in this case would be pyruvate, and a new amino acid, which would be glutamate. Glutamate is then shuttled to the liver, where it is acted upon by glutamate dehydrogenase. This interaction includes introducing water and NADP+, and it results in alpha-ketoglutarate, NADPH, and NH3, which is ammonia. This glutamate can also be acted upon by glutamine synthetase, which within the astrocytes of the brain will help to take up more of the ammonia from the tissues and create glutamine. Should glutamine remain in the brain, it can be dangerous as it is osmotically active. It means it can draw water into the brain and create cerebral edema. If left unchecked, this can create a coma, brain death, or brain herniation, or brain death. Glutamine that is returned to the liver is acted upon by glutaminase, which will free the ammonium and create ammonium and glutamate. This ammonium will then will combine with the ammonia and ammonium that were created in the initial reaction with glutamate dehydrogenase, and it will enter the urea cycle. How is nitrogen removal connected to gluconeogenesis? So during the fasted state, the body creates endogenous amino acids that can be then used for gluconeogenesis. So as mentioned above, when the glutamate interacts with glutamate dehydrogenase, it creates ammonia, NADPH, and alpha-ketoglutarate. Alpha-ketoglutarate can then enter the Krebs cycle and be used to create substrates that are eventually used in gluconeogenesis. Also, as listed above, when we do the transaminase reaction of alanine, pyruvate is created, which will also enter the TCA cycle and eventually be used for gluconeogenesis. In this patient, I initially had a very clear diagnosis of a deficiency in ornithine transcarmolylase, or OTC. However, in doing research of scientific journals and comparing the provided labs I have here, along with the differential diagnoses within the studies, I also have to consider a deficiency in arginase, and I'll explain why. I would say OTC deficiency is my most likely diagnosis in this child. I say this because the patient has significantly high ammonia level. This clearly shows that there's a defect in the urea cycle. The high levels of urine orotic acid are also due to a backup of the carbamoyl phosphate exiting the mitochondria, where it eventually creates orotate that will make it to the kidneys and be excreted in the urine as orotic acid. There's further proof of the defect in the OTC due to the fact that the patient's citrulline levels are low. This shows us that the carbamoyl phosphate is not combining with the ornithine via the OTC enzyme to create an adequate amount of citrulline. The fact that the deficiency, if this is in fact an OTC deficiency, arginine, which is usually a non-essential amino acid, will then become essential, which means it must now be ingested. Arginine is generally found in high protein foods such as red meat, fish. However, this patient's going to have to be on a very low protein diet, so they may have to consume this via a supplement. The reason I find myself questioning this diagnosis is in looking at research. OTC deficiency in neonates is generally only found in males, very rarely in females. It's also noted to have very low citrulline levels in the single digits. This patient, being a female and having citrulline levels still double digit, it led me to research an arginine deficiency. This patient doesn't fit that mold perfectly either, because arginine deficiencies very rarely has severely high ammonia levels like this patient does, and they generally don't have this high of orotic acid either. So, based on the knowledge I am going to assume OTC deficiency, but also a reasonable degree of suspicion for an arginase deficiency as well. In looking at this patient's lab values, we can clearly see, though, that the enzyme CPS1, or carbamoyl phosphate synthetase type 1, is functioning correctly. And we know this because regardless of which enzyme deficiency this patient ends up having, the patient is clearly producing carbamoyl phosphate. And switching to case 2, we'll start with scenario 1. This patient in scenario 1, the most likely enzyme defect is going to be that of branched chain alpha keto dehydrogenase, which is BCKD. When there is a deficiency of BCKD, the branched chain amino acids, which are valine, leucine, and isoleucine, along with their keto acid substrates, back up into the patient's system. There are no amino acids that are going to become essential because of this deficiency. However, the backup branched chain amino acids will inhibit the uptake of large amino acids, such as tyrosine and tryptophan, which in the brain, they are used to synthesize dopamine and serotonin. So it will inhibit some of their processes. It will inhibit the Krebs and ATP production via high levels of their ketone acids, which will encourage edema via inhibition of the sodium-potassium pumps, and it'll impair cell-to-cell communication by inhibiting GABA synthesis. Having a deficiency in BCKD is a disease otherwise known as maple syrup urine disease. It was named this as patients with this classic disorder are noted to have the odor of maple syrup in their urine, earwax, and sweat. There are 5 types of maple syrup urine disease, with classic, intermittent, and intermediate types being the most prominent. In the future, this patient is going to have multiple dietary restrictions, including a highly restricted protein intake. This restriction is due to the fact that branched chain amino acids are found in a myriad of foods like eggs, most meats, milk, soy, beans, nuts. The patient's also going to have a lifetime of blood draws to monitor plasma amino acid levels. Crises are easily brought on. Anything infection, stress, injury, even fasting for too long can cause a crisis in these patients. There are more aggressive treatments available, such as dialysis or liver transplant. However, these are rare and generally only seen in patients that are in crisis or with classic maple syrup urine disease. For the less severe and rare thiamine responsive type of maple syrup urine disease, thiamine supplements have been noted to increase the leucine tolerance, and it is also shown that very high doses of thiamine can potentially boost any residual branched chain alpha keto dehydrogenase enzyme activity. Moving on to scenario 2, I believe this patient is suffering from alkaptonuria. Alkaptonuria is a condition in which there's a deficiency in the metabolism of tyrosine. Patients with this genetic condition are born with it. However, visible symptoms are generally not present until they are well into adulthood. The first often missed sign of alkaptonuria is darkened urine. They say that it can even turn black if it's exposed to air for a few hours. But being that we generally get rid of urine so fast, flush it, throw the diaper away, this isn't something that is generally seen. So patients are usually into their 30s before they're going to start to notice problems with this. Things they might notice are thickened and irregular cartilage that can appear dark blue, gray, or black, and discolored sclera in their eyes. Also, and as in the case of this patient, they can note arthritis in their large joints, such as in their back or over their tendons, and other clinical manifestations include heart valve stenosis, kidney stones, and thyroid deficiency. All of these signs and symptoms are due to a loss of homogentate oxidase, which will cause an elevation in homogentic acid as a product of the tyrosine, which is a product of the tyrosine degradation pathway. Well, there have been trials of medications and vitamins such as high-dose vitamin C to help control the levels of the homogentisic acid, nothing concrete has been developed and treatment remains merely symptomatic, just treating people's symptoms. So there are also no amino acids that will become essential due to this deficiency. On to scenario 3, this patient would have been noted to have a deficiency in phenylalanine hydroxylase, or PAH, enzyme, also known as phenylketonuria. This means the patient is not able to convert phenylalanine to tyrosine, causing a backup of phenylalanine into the body. The effects of this backup can be seen within the brain. They cause intellectual disability, and if not diagnosed, treated immediately in infancy, can lead to death. These patients will also have their urine tested to ensure that this is in fact a PAH deficiency and there's not a defect in the B4 synthesis, which is a cofactor for the PAH. Treatment for PKU is initiated immediately upon diagnosis. The most common treatment is a low diet for phenylalanine for their life, with a goal of a level of less than 2 milligrams per deciliter. In current times, there are a few different options on how to control your intake. One treatment is to increase their intake of other large amino acids, such as tyrosine, tryptophan, leucine, etc. These large amino acids can become competitive inhibitors for the receptor sites that generally have a higher affinity for phenylalanine. There has also been success for some patients to take medications containing B4, which will stimulate any residual activity of the PAH enzyme. This would allow them to eat a less restricted diet, but still keep their levels low. There's also an enzyme substitution that can be found and approved in 2018, being phenylalanine ammonia lyase. This is a bacterial enzyme that breaks down phenylalanine to ammonia and tranexamic acid, which can then eventually be excreted in the urine. Regardless of which type of therapy is initiated for the patient, tyrosine will become an essential amino acid following this, as it is no longer produced in the reaction of phenylalanine with PAH and B4. Thank you.